Now, please welcome potato breeder and geneticist with the AAFC's National Potato Breeding Program, that's a mouthful, David DeCoyer, who will provide an overview of the AAFC's potato breeding program. Welcome, David.
Thank you, Derek. As Derek mentioned, my name is David DeCoyer. I am a research scientist at Agriculture and Agri-Food Canada, based in Fredericton, New Brunswick, but I run a national program which has activities all across Canada, particularly my breeding activities in New Brunswick and Alberta is the main focus. So today I'm going to give you an update of our breeding and genetics work. I want to acknowledge the Canadian Potato Summit for inviting me to give this presentation. In today's presentation, I will talk about potato breeding in the AAFC. I will discuss breeding strategies to meet AAFC's strategic mission, and then I'll discuss briefly some new varieties and selections that are available for testing. So through the breeding continuum, there are many steps. We work with germplasm collections. We develop new knowledge and enabling tools for crop development, which includes molecular genetic research and breeding research, new breeding techniques. This continuum advances to the germplasm development, where we characterize different traits and develop new strategies for breeding. And finally, through the incorporation of new traits by breeders, we enter into cultivar development in collaboration with industry, and AAFC is involved in different levels of these activities and is supported by many other disciplines. In the past 5 years, we've been working on updating our breeding pipeline. So the breeding process developed is initiated through hybridization to generate botanical seed, and then these seeds are grown in the greenhouse, and then from those tubers are produced. They go to the first year and are grown in our breeding locations in Benton and Vauxhall. Starting in year 2, we start to multiply our seed and then start to do evaluations. And in years 3 and 4, we enter into preliminary trials and start to incorporate DNA markers into the research and breeding. And finally, in years 5 and 6, we enter into national trials across the country with many of our collaborators. Once a good selection is identified, these selections go into industry trials and eventually are released as varieties and introduced into the seed system. As I mentioned, we have national sites across Canada, from British Columbia all the way through to Newfoundland, with our primary activities being conducted in New Brunswick and Lethbridge. So this slide just shows a different more details about the activities that occur at each of the sites in Alberta, Manitoba, Ontario, New Brunswick, Prince Edward Island, Quebec, Saskatchewan, and BC. And I want to highlight that we're working with, have some work in Newfoundland on potato wart resistance. So recently, in 2022, Agriculture and Agri-Food Canada developed a strategic plan for science. This involved 3 pillars. The first pillar is mission-driven science. Second pillar is people first strategy, and the third pillar is organizational excellence. And our work is divided into 4 mission areas. The first one is related to climate change. The second one is related to resiliency of ecosystems. The third mission is about the circular economy. And the fourth is about digital transformation of the agriculture sector. So related to our first mission, mitigating and adapting to climate change, we're work in our potato research is targeted towards climate adaptation solution, and elements of our work in this area include improved stress tolerance and adaptation to climate change. Many aspects of stress tolerance come into play here. As the picture shows, one is the reduction in defects that are caused by heat and drought stress. The other aspects are related to quality, and of course, under stress, you can have reduced yield. Another aspect of this mission is to enhance sustainable food production in the north, and one strategy that we're using to target this area is looking at earlier potato varieties and true potato seed that might reduce the shipping costs and give that region some advantage in the future as climate change continues to affect that region. So a critical element of meeting our goals for this mission are the national trials. Through these trials, we test breeding selections across the country in diverse locations to compare their performance under different conditions. These conditions differ in the amount of the soil types, the type of and availability of water, the temperature, the length of the growing season, and they're subject to these trials are grown under the regional production practices. And in this diverse trials, we're able to assess resilience and performance under this climate stress. Another purpose of this importance of these trials is to advance selections in the product development cycle. And as we get data from across the country, we get far greater confidence in the data that will be used to support the release of a variety. And these trials also provide industry members opportunities to participate in selection decisions and give feedback on clones. And we want to engage with the users and to ensure that our selections are relevant to meeting their product profiles. The second mission for AAFC is increasing the resilience of agri-ecosystems. And there are several aspects to this mission, but highlighting 2 of them. One would be to enhance the biodiversity to stimulate productivity and resilience. And in this case, we're leveraging the potato genetic resources that are available to develop resilient crops, such as disease resistance and abiotic stress tolerance. We're also looking to reduce the impacts of pests and diseases through really biovigilant approach, and then also involves resistance to biotic and abiotic factors. Recently, as you all may have heard, potato wart has been a very important disease in Canadian agriculture, and we're trying to enhance the number of varieties with potato wart resistance through collaboration with with partners in Newfoundland, where the disease is endemic, and we're able to do field testing. On this slide, you see some examples of the the warty symptoms, but we also have identified resistant selections through this testing, and we're enhancing our capacity for this type of research in Newfoundland through improving our trial site in Avondale and conducting this more potato wart research at the St. John's Research and Development Center. We are in our breeding program, we look at many different diseases. These include PBY, PBX, golden nematode wart, late blight, fusarium dry rot, common scab, verticillium, and to a small degree, Colorado potato beetle resistance. So our goal is to increase the frequency of these resistances available in our advanced selection, and we're making progress. It's kind of a incremental progress, I would say, but every year we have more selections available with these tolerance attributes. The third mission is one about advancing the circular economy. This is about reducing food waste, but it also entails diverse production systems with multiple outputs and co-benefits. And of course, you all know that potato is a very diverse crop, and we have an opportunity to take advantage of emerging food and bioproduct opportunities. This includes colored fleshed potatoes, such as those illustrated on this slide. We're also looking to enhance the quality and longer shelf life and reduce food loss, primarily through maintaining and having good product quality and improved product recovery through storage. So looking at having varieties with fewer defects, those are able to store for longer time periods at lower temperatures and have resistance to storage pests and diseases, such as fusarium dry rot or other soft rots that could be associated with the growing season. The fourth mission is accelerating the digital transformation of the agricultural sector. To do this, the department will work towards enhanced data-driven agricultural systems. And in our research, we'll be adopting data science fundamentals for breeding and germplasm development. We'll be increasing our application of genomics and open omics data science and digital dexterity, which basically means that we're trying to use all the available tools that help us gather objective data to help make our breeding more efficient and effective. We're also going to increase the visibility of our collected data as a way to increase collaboration with other research institutes as well as the potato industry. Data is critical along the potato breeding continuum. We are working to optimize our operations and create new opportunities, and this applies from genomics to commercialization and of course all of the operational aspects of breeding. So some examples of the places where data is important includes making decisions on which crosses to make, which seeds to plant in the greenhouse, to generate our next generation of breeding material. During the growing season, data is used to help make selection decisions. As the selections go further on in the breeding cycle, we get enhanced, or our data amounts grow exponentially, and we are relying more and more on on computers and data analysis to help make our selection decisions for that material that is going into regional and national trials, and ultimately into commercialization. So we have developed a breeding databases that work along the continuum of our breeding program. So we record all the information related to our crosses, our families, and the individual, starting at the individual seedlings, all the way through to trials, and data collection and molecular data that we include in our data analysis. We collaborate with different institutions to that have developed useful apps that help us collect the data, particularly some universities in the U.S. We are developing tools for faster reporting and objective data collection. We're using a lot of image analysis. We've developed a database, as I mentioned, called Potato Base, that is used in our program extensively. And at our two main breeding locations and in Charlottetown, we have optical graders that allow us to collect data on individual tubers in each plot, which gives us some advantage to look at the uniformity and distribution of the tuber size and shape profiles from each selection. As we advance in over time, we're using more and more methods to collect trait data, and these methods generate a vast amount of large data sets. Some of the examples, as I mentioned, one of them was the optical sorter or data collection, but we're also collecting data from images and different spectral measurements we take either on individual tubers or by using drones or other methods to capture the above ground portion, and we're also have in the future expect to be looking underground using different technology as well, which I think will be very valuable for our decision making. The application of these tools allows us to perform genomic analysis in our breeding material. And one example that we've used when we combine data from DNA with the data from our trials that we collect in the field has led us to identify very strong quantitative trait loci which impact, for example, in this case, yield. So on chromosome 5, we've identified a location which will reduce or conversely increase yield based on our information, approximately 50 hundredweights per acre. And just to illustrate some of this, how we show this on a graph here, we have so potato is a tetraploid. It has 5 different genotypes. I don't know if I can oops. 5 different genotypes. So on the top left is one. We have 4 A's, and the bottom left 4 B's, and there's 3 heterozygotes in the middle, and you can kind of see a trend line from going from a total yield of about 430 hundredweight per acre to less than 250. So this kind of demonstrates that, and these points show the average of all of our material that have this particular genotype at this locus. So when we overlay this with some of our breeding materials, we see that we have a pretty good relationship between the expected and actual yields. So particularly for some, our russet Burbank is slightly above the average. Epity is about the average, and in some cases, like caribou, is quite higher than the average. But this, shows that this is not a silver bullet, but it gives us an overall perspective of what genetically is needed for a variety to have a very high yield. We know that there's many genes involved, and we're continuing to look at kind of an overall genome-wide approach to identify and use the genome-wide information to predict which parents and which selections will have the best performance that allow us to make our breeding targets, like high yield, excellent quality, disease resistance, etc. And this busy slide, I just wanted to emphasize that this information then can be used to allow us to optimize our parental and mating allocations. So how many times would you use a particular variety or selection in your crossing block, and then how many seedlings you might plant out in the first year from each family. So a combination of support from industry has allowed us to release several varieties recently. I have some of these listed on this slide, and I'll just quickly go through some of them. We have AAC Eastern Russet, which is a russet with yellow flesh, blue sapphire, which is a purple, Santa Margarita, which is a red skin, yellow flesh, and AAC Odyssey, which is a red skin, red flesh. We have others, more recent ones: Robin, Canada Gold Doré, Griffin Russet, Caddy, and Africa Caddy, and then we have some Midnight, Commander, Arctic, Crimson Tide, and Brava. And the most recent ones were Red Fox, Garnet, and Mulberry. And I believe I'm very close to my time, so I'd be happy to. I would like to acknowledge all of our technical team and farm support in AAFC in Benton, Fredericton, Lethbridge, and Vauxhall, as well as the collaborators on our national potato variety trials, and the funding that we received from AAFC and industry through the Canadian Agricultural Partnership Program. And thank you for your attention. I have contact information here, but I'd be happy to answer any questions that you might have.
Thank you very much for that, David. That was much appreciated. Yes, we do have some little bit of time here for some questions. I do see one of them in the Q&A thing that I can ask you right now. In your actual and predicted yield of varieties, can I assume that King Russet would be comparable to caribou?
So we didn't include King Russet in this study. I know it's a private variety, but it would really depend on the genotype. But we know that from my understanding, it's a very high yielding variety. And part of what I wanted to illustrate in there is I think breeders are doing a good job of selecting those varieties that are kind of above average for what their genetics might predict. So as we learn more and more, I think that we'll get closer to being able to match that prediction with the actual yields, and balance that. Maturity is strongly linked to yield. So in Canada, we know that later maturity often causes some problems if we have an early winter.
Perfect. And I actually have one question for you, David. Are there any particular varieties of potatoes that are more resilient when it comes to climate change, and vice versa? Are there varieties that are more susceptible? And of those varieties, what traits make them resilient or susceptible to changing climate?
Well, it's a very good question. I think if we knew all of those answers, we would be able to do much better job at breeding. But it's partially, part of the issue with breeding for these traits is they're a bit unpredictable and kind of after the fact. I think you start to learn about what those traits are that help lead to those resilient traits. So I'm not going to recommend any particular variety, but I think as you learn about these traits through the type of trials that we're doing now over diverse locations, many years, and it just emphasizes the importance of this type of work to be able to come up with a picture that is kind of not just a regional one, but a national one, and then layer that national perspective over several years. So I think the importance of this work is hopefully recognized through this snippet of information that I've given you today.
Perfect. Well, we do appreciate it, David. Thanks for joining us today.
You're welcome.